Welcome back, everybody. Today, we're going to be diving deep into the world of Class C G protein-coupled receptors.
The GPCRs?
Yeah, GPCRs for short.
Right.
And specifically, we're going to be focusing on metabotropic glutamate and GABA receptors.
Okay.
You're interested in how these receptors could revolutionize neuropsychiatric treatments. And that's exactly what we're going to explore.
Sounds good.
So
Yeah.
to kick things off,
Yeah.
what sets Class C GPCRs apart from other receptor types?
Well, picture this, a Venus flytrap just waiting for its prey. That's essentially what the extracellular domain of a Class C GPCR looks like. It's this unique structure that sets them apart.
A Venus flytrap.
Yeah.
Okay, I like that visual.
Yeah.
So how does this Venus flytrap mechanism actually work?
It's all about binding. When a molecule like glutamate or GABA comes along, it binds to this Venus flytrap domain, causing it to snap shut. This triggers a cascade of events inside the cell, ultimately altering neuronal activity.
So it's like a lock and key, but with a molecule triggering a cellular chain reaction. That's pretty cool.
Yeah.
And this is what makes them such attractive drug targets, right?
Exactly. If we can design drugs that precisely fit into that Venus flytrap domain, we can potentially modulate the activity of these receptors and influence brain function. Interestingly, the first GPCR structure ever crystallized was rhodopsin, a protein found in our eyes that helps us see.
Wait, so the structure of a protein in our eyes helped scientists understand receptors in our brain. That's fascinating.
Yeah, it highlights the interconnectedness of different systems and how fundamental discoveries can have ripple effects across scientific fields. The rhodopsin breakthrough really paved the way for targeting GPCRs for therapeutic purposes.
Okay, cool. So you mentioned glutamate and GABA receptors. Can we break down the differences between these two?
Sure, they both belong to Class C GPCRs but have distinct roles. Think of glutamate receptors as the accelerators of neuronal activity. They make neurons more likely to fire amplifying signals in the brain.
Okay, glutamate gets things going. What about GABA?
GABA receptors are like the brakes. They inhibit neuronal activity dampening signals. It's this balance between excitation and inhibition that allows the brain to function properly.
Like a carefully orchestrated dance.
Exactly.
This makes me think about conditions where this balance is disrupted, like epilepsy. You mentioned earlier that Class C GPCRs are implicated in a wide range of neuropsychiatric conditions. Can you give some specific examples?
Absolutely. Take Mgular R23 agonists, for example. Studies have shown that activating these receptors can reduce seizure frequency and severity in animal models of epilepsy. It's like fine-tuning the brain's braking system to prevent those runaway electrical storms that characterize seizures.
That's really promising. Are there other conditions where targeting these receptors shows potential?
Definitely Mgular R5 antagonists, which block a specific type of glutamate receptor, have shown promise in treating symptoms of Parkinson's disease. It's like a workaround targeting a different receptor to ultimately influence dopamine pathways, which are disrupted in Parkinson's.
So manipulating one type of receptor can have cascading effects on other pathways. That highlights the complexity of this system. But how do we know where to target? I mean, the brain is a vast network.
You're right. Location is key. While these receptors are widely distributed throughout the central nervous system, their concentrations vary in different brain regions. This uneven distribution is crucial to their specific functions. For instance, those MGLR5 receptors we talked about are found in high concentrations in areas involved in movement control, which makes sense given their potential role in treating Parkinson's.
So it's not just about the type of receptor, but also where it's located in the brain. This is getting really intricate.
It is, but that's what makes it so fascinating.
Shall we delve deeper into the signaling cascade next? I'm curious to know how that doorbell ringing actually translates into a change in the cell's behavior.
Let's do it. Okay, so let's imagine a bustling city. The receptor is like a busy mailbox, constantly receiving messages in the form of molecules like glutamate or GABA.
Okay, I'm with you.
Now, when the right molecule, let's say glutamate, arrives at the mailbox, or in our case, the Venus flytrap domain of the receptor, it triggers a series of actions inside the cell.
So it's not just passively receiving a message. It's actively setting things in motion.
Exactly. Think of it like a chain reaction. The first step is the activation of a G protein, which is a molecule located inside the cell right next to the receptor.
So like a messenger waiting for instructions.
Precisely. The G protein now activated doesn't just stand around. It interacts with other molecules, triggering a cascade of events. These events can lead to changes in the activity of ion channels or enzymes, which directly impact how the neuron fires.
So it's a multi-step process with each step amplifying the initial signal.
You got it. It's a symphony of molecular interactions, all orchestrated by the initial binding of the neurotransmitter to the receptor.
Now, I remember you mentioning a protein called GRK2, which is involved in regulating this signaling cascade.
Right.
It's like a behind-the-scenes director making sure the show runs smoothly.
Yeah. That's a great analogy. GRK2 plays a role in desensitizing GPCRs, essentially turning down the volume on the signal after it's been received.
So it prevents the cell from being overwhelmed by constant stimulation.
Exactly. This desensitization process is crucial for maintaining proper cell signaling and preventing overstimulation. What's fascinating about GRK2 is that its role extends beyond the nervous system. It's also involved in cardiovascular health and pain modulation.
Really? So a protein involved in fine-tuning brain cell communication is also linked to our heart and how we perceive pain.
It highlights the interconnectedness of different systems in our bodies and how seemingly unrelated processes can share common molecular players.
That's pretty amazing. It makes me think about the potential ripple effects of targeting these receptors. Could manipulating GRK2 in the brain have unintended consequences for the heart, for example?
That's a crucial question that researchers are actively investigating. Understanding these complex interactions is key to developing safe and effective treatments.
Right. It's all about finding that sweet spot, that precise balance.
Yeah.
Now let's shift gears a bit and talk about GABA receptors.
Okay.
I know our listeners are particularly interested in their unique two-subunit structure.
Of course, GABA receptors are fascinating because they're heterodimers, meaning they require two different subunits to function. It's like two pieces of a puzzle that fit together to form a functional receptor. We call these subunits GABA1 and GABA2.
So they need each other to get the job done. What happens if one of the subunits is missing?
The receptor simply won't function. Each subunit has a specific role to play. The GABA1 subunit is the one that binds to GABA, triggering the initial doorbell ring. The GABA2 subunit, on the other hand, is crucial for interacting with the G protein inside the cell.
So GABA1 is like the receiver and GABA2 is the translator relaying the message inward.
Exactly. A perfect analogy. It's a beautiful example of molecular teamwork with each subunit playing its part to ensure proper signaling.
This complexity is fascinating, but it also makes me wonder about the challenges of targeting these receptors for therapeutic purposes. If we need to manipulate both subunits, does that make drug development more difficult?
It definitely adds another layer of complexity. Researchers need to consider how drugs might interact with each subunit and whether targeting one subunit over the other might have different effects. It's a puzzle, but one that scientists are actively trying to solve.
And I bet our listeners are starting to see why this is such a cutting-edge area of research. There's so much we're still learning about these receptors and their potential. Now, before we dive into the therapeutic landscape, I want to touch on one more thing. Synaptic plasticity.
Okay.
I know this was something you highlighted as particularly interesting about Class C GPCRs.
It is synaptic plasticity is the brain's ability to change and adapt over time. It's the foundation of learning and memory. And guess what plays a crucial role in modulating these changes? You got it. Class C GPCRs.
So they're not just static players. They're actively shaping the connections in our brains.
Exactly. They can influence the strength of connections between neurons, making them stronger or weaker, depending on the context. This dynamic interplay is essential for everything from forming new memories to adapting to new environments.
That's amazing. So by targeting these receptors, could we potentially enhance synaptic plasticity and improve cognitive function?
That's a very exciting possibility that researchers are exploring. There's evidence to suggest that activating certain Class C GPCRs could promote the formation of new synapses, potentially leading to improvements in learning and memory.
That opens up a whole new world of possibilities.
It does. Think about the potential implications for conditions like Alzheimer's disease, where synaptic loss is a hallmark. If we could find ways to boost the synaptic plasticity, we might be able to slow down or even reverse cognitive decline.
Wow, that's incredible. Like rewiring the brain to combat disease. Now, I know our listener is eager to hear more about the therapeutic landscape. Can we dive into that next? I'm particularly interested in the specific areas of focus in drug development.
Absolutely. Let's explore that in more detail. So we've established that Class C GPCRs are implicated in a range of neuropsychiatric conditions and that their role in synaptic plasticity makes them particularly intriguing targets.
Yeah, for sure.
And the therapeutic landscape is rapidly evolving with several key areas of focus right now.
Like what? Can you give us a roadmap of where the research is headed? I know our listener is keen to understand what the future holds for these potential treatments.
Absolutely. One major focus is developing drugs that are more selective, targeting specific subtypes of these receptors instead of the entire receptor family.
So rather than going after the whole family of receptors, they're trying to be more precise.
Exactly.
Would that potentially minimize side effects?
That's the hope. By honing in on specific subtypes, we can potentially achieve greater therapeutic efficacy with fewer off-target effects.
It's like using a scalpel instead of a sledgehammer.
Precisely.
Oh.
Another exciting area is exploring new drug delivery methods. Imagine if we could deliver drugs directly to specific brain regions affected by a particular condition.
That would be game-changing. I'm picturing tiny nanobots carrying drugs to their precise targets.
It's not science fiction anymore. Researchers are actively developing innovative delivery systems that could make treatments more effective and less invasive.
This is all incredibly promising. It seems like we're on the cusp of a new era in neuropsychiatric treatments.
We are, and Class C GPCRs are undoubtedly at the forefront of this revolution. Their unique structure, their intricate signaling pathways, and their involvement in synaptic plasticity make them prime candidates for drug development.
It's clear that these receptors hold immense potential.
Absolutely.
Now, before we wrap up, I want to leave our listener with a final thought-provoking question. Given the wide-ranging roles of Class C GPCRs in the brain, what other unexpected connections might we uncover in the future? Could there be links to conditions we haven't even considered yet?
That's a great question, and it highlights the beauty of scientific inquiry. We're constantly discovering new things about these receptors and their functions. I think it's entirely possible that we'll uncover links to conditions we haven't even thought of yet. For example, there's emerging research on the role of GPCRs in the gut microbiome.
Wait, the gut microbiome. How are those two things connected?
Well, it turns out that the gut and the brain are in constant communication through a complex network called the gut-brain axis.
So it's not just a one-way street. What happens in the gut can impact the brain and vice versa.
Exactly. And some of these signals are mediated by GPCRs located in both the gut and the brain.
Wow. So could imbalances in the gut microbiome potentially influence GPCR signaling in the brain and contribute to neuropsychiatric conditions?
That's a hypothesis researchers are actively exploring. It's a fascinating area of research, and it really underscores the interconnectedness of different systems in the body.
That's really interesting. I'm starting to think that we're just scratching the surface of what we know about GPCRs.
I agree. There's a whole universe of discovery waiting to be explored, and I have no doubt that Class C GPCRs will continue to surprise us with their versatility and therapeutic potential.
Well, I think it's safe to say that our listener has gained a whole new appreciation for these remarkable receptors. We've covered a lot of ground today, from their unique structure to their intricate signaling pathways to their potential for revolutionizing neuropsychiatric treatments.
It's been a pleasure discussing this fascinating topic, and I encourage our listener to continue exploring and asking questions.
Until next time, keep diving deep into the world of science and never stop learning.
Farewell and happy exploring.